Good morning, students. Today we are going to discuss the role of cytology in taxonomy. So first we'll have a brief introduction about taxonomy. It is a science that deals with the study of identification, nomenclature, and classification of plants. As classical taxonomists, they believe that we can easily identify and classify plants on the basis of morphological characters mainly. So they believe that morphological characters are enough to place a particular organism in some group under the hierarchical system of classification. But till 20th century, this has been established that alpha taxonomy, which mainly deals with the description, the morphological description of the organisms, that is not adequate for identification and classification of the plants. So as a result, taxonomy entered a synthetic phase. Means now we are not only talking about morphology, but we are getting the information from other fields of science as well, so that they are given due consideration while classifying and naming a particular organism. Now, one such field which is being utilized for this process is cytology. Now, what information from cytological data can be deduced? We can have comparison on the basis of number of chromosomes, their size, their appearance, bending patterns, aberrations, behavior of chromosomes when they are separating during meiosis. So all these things, they can give a reference whenever we have to characterize a particular organism, or we have to promote it from a lower level of the taxonomic hierarchy to the higher level. All these attributes of the cytology are very, very important. Now, how cytology is related to taxonomy? Cytological studies of flora have always been helpful in finding out the evolutionary status of plants. Now, how they are helpful? It has contributed in the form that now we can compare chromosome number, chromosomal size, and the behavior of chromosomes during meiosis, which can be easily used to distinguish different genera, species, subspecies, tribes, etc. So we can have a clear-cut differentiation of the organisms of different ranks. So because of such an immense contribution of cytology to taxonomy, a new discipline has developed which is called cytotaxonomy. So these are once again the three parameters on which the cytotaxonomy is based. Now first is chromosome number. For any taxon you all know these are the different levels of the taxonomic hierarchy. Any particular level is called a taxon. That may be a family, order, genus, class, species, etc. So for any taxon, the basic chromosome number is normally constant. So that helps in taxonomic grouping of diverse group of plants. So even once you observe a particular species, for example, you found species X, Y, Z, and you observe that here the base chromosome number is 5, here it is 6, here it is 5. So it clearly indicates that these two species are related. So you can keep them under the same group. Then larger the number of chromosomes is considered to be primitive character, and the groups or the taxons which are having small number of chromosomes, they are considered advanced. So this indirectly helps in placing a particular group before or after in the hierarchy. Second is size and shape of chromosomes. The size of chromosome is highly variable. We can have their size varying from 0.5 to 30 microns. In higher plants, when we observe, there is a lot of variation in the size of chromosomes, and this is normally associated with the morphological specialization, and it can even provide the information regarding the evolutionary tendencies within the taxa. So this has been observed that larger the chromosomal size, that has lesser number, and such plants, they are advanced. For example, wheat, rice, these are grass species which are kept under the family Poaceae, which is an advanced family. They have less chromosome number but large size. So this is considered an advanced feature. Third is the chromosomal behavior during cell division, that is during meiosis. So when the chromosomes are going to separate, then sometimes we observe some kind of the abnormalities during that phenomena. So those abnormalities, if they are found common to some particular taxon, they can be kept together. So these may be non-pairing of the chromosomes. There may be change in size of chromosomes during meiosis, inversion, translocation, that may also be there. Then laggard formation. Laggard is a chromosomal segment without centromere. There is formation of bridge, which is the most common one. You can observe here the chromosomal bridges, they can be seen. Here we can see a laggard, which is a acentric fragment, so it cannot place itself on the spindle apparatus. Then early or late disjunction or non-disjunction. That means when the homologous pair of chromosomes during anaphase they fail to separate, that is called non-disjunction, or there may be certain delay in their separation. Then duplication or deletions. So all these, if some abnormalities, some similarities observed in the abnormalities, then those organisms they can be placed together. So these were the three main aspects which are actually used for cytotaxonomy. Now, what is the significance of cytotaxonomy? That now we are going to quote different examples from the past where the cytotaxonomic studies have actually helped in taxonomy of the genera. So the first is at family level. So karyotype studies, they are very helpful in differentiating the organisms at the angiospermic family level. For example, we have two families, Viscaceae and Lawrenthaceae. Earlier they were placed together, but when we observe their chromosome number, Lawrenthaceae has base number 9 and Viscaceae members they have base number 10 to 14. That clearly gives an indication that these organisms they cannot be placed together, so they should be assigned the separate family status. Second is at the subfamily level. So subfamily, here again we have the example of Gremini. Gremini has number of tribes, but they can be easily arranged on the basis of the cytological data. Third is at the genus level. So chromosomal studies, they have been used for different genera like Lewis et al. He has traced the lines of evolution in Clarkia, and all species of Clarkia, they have been merged with Godatia because they were having cytological resemblance. Similarly, agave and yucca, earlier they were placed under separate families, agave under Emeraldaceae and yucca under Liliaceae. But when we observe their chromosomes, they have the same type of the chromosomes, that 5 are long and 25 are short. On that criteria, they have been placed together under the family Agaveaceae. Then at species level also cytotaxonomic studies have been very important. Like Lewis et al. Has studied Clarkia, Hutchinson et al. Studied Gossypium, and they have been able to mark the different species on the basis of cytological studies. This is Clarkia. This is Gossypium. You can see number of species are there, so these species can be easily distinguished on the basis of cytology. The next is at the subspecies level. Now, chromosomal studies, they have proved useful in delimiting subspecies and varieties in many families. Here we have observed that within the same genus and the species, there are different groups which are having different cytological behavior. Here we have an example. Sharma has actually suggested that these cytological races, they represent the evolutionary stages. Means let's suppose the different organisms at present stage they are having some cytological differences. With the passage of time during the evolution, ultimately they may get a status of subspecies or they may be promoted to species. That is how the course of evolution takes place. So all these, they are the phases of evolutionary stages, and these cytotypes, they whenever they acquire sufficient morphological variations because of their cytology, they can be raised to different variety status, subspecies or species level. So here is an example of Veronica prostrata. This has two further subspecies, prostrata and schiri, you can see here the base number is different. Depending upon that base number, if the morphological variations also arise in future, they can be easily get promoted to the species level. So these are the two, Veronica prostrata and Veronica prostrata schiri. Now, you can also have the observation about the chromosomal lengths. Here, this is the Allium cepa which has been cultivated in the field. You can see the chromosomes here. Allium cepa, we have total 16 chromosomes in this. And whenever the cells they are raised from the somatic embryogenesis, that is under tissue culture conditions, you can see the length of the cells have increased. So overall behavior, depending upon what kind of the mode of the development they had, then also you can easily observe the cytological variations in the cells. So we can conclude from this discussion that cytotaxonomy has emerged as a very strong field of research as far as the categorization or the identification of different plant angiosperm families are concerned. Then chromosomal data is very strong tool for the correct interpretation of the phylogenetic relationships and evolutionary tendencies in different groups of plants. So on the basis of this chromosomal data, just by comparing the base number or their size or their behavior during meiosis, we can easily find out whether a particular genera is related to the other one or not. And we can also find out what can be the course of evolution, whether it's an advanced genus, it has to be kept under some advanced group or primitive group. So that is how cytotaxonomy is a very, very powerful tool in the field of taxonomy. Now, just giving you a brief information about karyotyping, because whatever studies are being carried out in cytotaxonomy, they are based on karyotyping. So, karyotyping is a study at mitotic metaphase. So, we select the chromosomes at metaphase when they are maximum condensed and shortest of their length. Now, how do we define karyotyping? This is a phenotypic appearance of the somatic chromosomes or the basic set of the somatic cell at metaphase as seen under light microscope. So, we are going to click the cell at metaphase, and then those chromosomes are arranged in the decreasing order of their length, and then we compare a karyotype of one organism with another organism. So what is the basic procedure? We take plant root tips, then we treat it with colchicine. Colchicine that is going to disrupt spindle. So because of that, there will be no spindle formation and chromosomes won't be able to align themselves on the spindle apparatus. So the number of the chromosomes that will get doubled, but there will be no further separation towards opposite poles. Then we are going to stain those chromosomes with acetocarmine, that is a chromosomal stain. The slide by squash technique, the slide is prepared. Then we observe the cells at metaphase and we click the photograph of those particular areas. Then chromosomals, they are cut along their length and they are arranged. Those pictures are arranged in the decreasing order of their size in homologous pairs. So first you have to trace the pairs. For example, let's see, these are metacentric. This is the picture that we obtained at metaphase. Since it is after the treatment of colchicine, this is called C mitosis, C metaphase. So then we are going to cut each segment of the chromosome, and then we will match depending upon the length or position from the centromere. We are going to make homologous pairs, and those pairs, the overall on the basis of length, we start from the longest one to the shortest one. This is how the karyotype is prepared, and then we can easily compare this karyotype for different organisms. Okay, so all cytological studies, they are based upon karyotyping. So that's all for today's lecture. Thank you.